Online. You guys, hello, those who are tuning in, the nine that are watching, welcome to one of many streams, one of many free webinars on this channel. So, hello, those who are tuning in. That was an echo just then. So, yes, today's plan is this: key ideas that overlap, that ties in with photosynthesis, ties in with respiration as well. So that's one of the goals today. Additionally, we're going to go over biological molecules. We're going to go over enzymes, a bit of Corm's guidance, transport processes, circulatory system, and the use of, the understanding of what is found in blood, the blood components, lung mechanics, endocrine system, neuronal coordination, and ecology in one slide. So that's today's plan. I'm just going to wait for if there's a bit more of a turnout. It's 8 people watching. If the minute it hits, let's say 10 to 15, maybe, then I'll go ahead with the stream. Okay, so just we'll give it a few more minutes. We'll start, in fact, let's say in the next 5 minutes or less, maybe. Let's say 11:07. We'll make a start. And also, if you could let your peers know of this live stream, because we are potentially going through we're looking at probably 20 lessons, even a whole year of content in possibly like less than 2 hours. It would be a waste of time if oh well, it would be can't really phrase it, can I? But it would be a waste of an opportunity. Okay, just 2 more minutes, then we'll go ahead with the program. So yeah, 11:07. We'll make a start. Okay, there's going to be like 3 or 4 questions. So I'd recommend you have your notepad with you and pen and jot some ideas whilst we go, of course, go through content. Okay, let's just make a start. So key ideas that overlap. Now, the big bioenergetic processes include photosynthesis and respiration. These 2 key ideas are prevalent throughout many different topics or many different diseases. Like with photosynthesis, it ties in with crop yield, which is in topic 5, the use of biological resources, the use of greenhouses. So if you create let's say, mimicry of light and CO2 concentration, you're going to maximize the photosynthetic rate and therefore encourage growth. And with respiration, the whole goal of respiration is to release energy slash ATP. And as you know, guys know, ATP is of course, it shares the same name as the Tennessee governing body, but also it's the energy currency we sort of give that name for it. In A level, you would be using ATP rather than energy. And those who are planning to do AQA A-level biology, there's a 25 marker essay, and it's fundamental you refer to A-level nomenclature. But that, of course, we've got GCSEs next week. Let's figure out what A-level bio for 1 second. Going back to this, you have key ideas. You have bioenergetic processes that include photosynthesis and respiration. Let's actually write the word equation for photosynthesis. You have CO2 plus water plus C6H12O6 plus oxygen. Okay, I'm not going to bother about balancing because obviously we've got to go through a lot today. But you know the main goal really is to produce glucose, and glucose links with growth. Now glucose can be converted into a bunch of other things. Glucose can be converted into amino acids. I know my YouTube channel is called amino acid, but I don't want you to make that mistake. Please spell the word acid correctly. And of course, amino acids joined together make a polymer of proteins. Now, glucose can also be converted into cellulose. Now, cellulose is found in the cell wall. It's this polysaccharide substance that provides the cell wall with structural integrity, and it's insoluble for one, so that process of osmosis does not disrupt it, the sort of stability of the cell. Another thing that glucose can convert into is starch, and starch is a energy storage. So crucially, glucose can convert it into amino acids, protein, cellulose, starch, and so on and so forth. It can also be converted into, well, be stored as cell sap. But the go-to really in your mark scheme, if you want to be a mark scheme merchant, is to use, and if the question's tying in with growth, you would talk about amino acids and proteins as glucose converts into those things, and as well as cellulose. These tie in with growth. So let's say you had a disease called tobacco mosaic virus, and it affects the light, the ability of chloroplasts to absorb light. So let's say it reduces chlorophyll production, or really damages it, discolors it, then you would have less photosynthesis occurring. But the question, let's say, explain why tobacco mosaic virus causes the stunting of growth. Well, you would say less, less, less. So less photosynthesis, less glucose being produced. Okay? Even crucially, as I didn't mention here, light is absorbed. Photon of light is absorbed in photosynthesis. Chlorophyll absorbs it. So mention that remark, less photosynthesis, less glucose, less amino acids, less proteins can be generated, all of the above. Okay, so that's one of the key ideas that can tie in with photosynthesis. Photosynthesis can promote the quality of a product, quality of a agricultural product, so on and so forth. And it's important to understand that. So you have growth that can cause photosynthesis, and then you have so photosynthesis causes growth, and then you have energy released by respiration. And let's go to an empty slide. We've got an awful lot to go through. So respiration, there's two types, of course. Aerobic respiration. Aerobic respiration uses oxygen. Okay. Aerobic respiration uses oxygen, and the following word equation demonstrates this, because obviously it's on the reactant side. Glucose and oxygen goes to make carbon dioxide plus water, and crucially in it, inverted commas, energy slash ATP. Okay. Now this topic, respiration, links with one of the slides in today's session, and it's to do with blood, isn't it? Red blood cells. Now, the link you can draw is the fact that red blood cells carry a protein called hemoglobin. And then that hemoglobin affinitizes or binds to oxygen molecules. Of course, oxygen is a key player, isn't it, in this aerobic respiration pathway reaction. And when hemoglobin binds to oxygen, it and the red blood cells roam around in the blood and then give it to, let's say, a working muscle. In this case, everyone's brain regions at the moment, if they're concentrating on the stream, then they're going to unload that oxygen for those brain cells, and then those brain cells can undergo this process called respiration, and typically aerobic respiration, and you can release energy. So there's a lot of links you can draw, especially with those circulatory system problems or ways to improve, let's say, energy and ways to improve performance in a race. So one example I can think of is Lance Armstrong. He used EPO. And what EPO did was it increased red blood cell count, which in essence increases more hemoglobin and oxygen binding, which in essence increases the respiration rate, which in essence increases more ATP, more energy, less fatigue, so on and so forth. So that's a key idea. So yeah, so that's done now. Bullet point number one. So then we'll start off with biological molecules. So I'm not sure what different schools have, but maybe you're starting GCSEs in year nine, maybe you're not. But point being is that this is a topic that will come up in the test, obviously, and biological molecules, and it shares something in AQA as well. It's one of the first topics on the AQA course, A-level course that is, and you have the big three. So the big three include proteins, okay, and it's important to understand. There's so many examples. It's important to understand the examples of proteins. So, for instance, you have the one I just mentioned, hemoglobin. That's an example of a protein. Okay. Other examples include enzymes, which ties in with today's session as well. They're a protein. Okay. Other examples include some hormones, like peptide hormones, such as insulin. And can anyone tell me what insulin does? Well, what insulin does is reduce glucose levels, and I think I mentioned it in my gene modification video. So, if you're still struggling with genetic modification, it's important that you watch that video. So, you've got peptide hormones such as insulin, amongst other proteins. You have, I think, antibodies, of course. Antibodies are a chief example of a protein. The thing that are secreted by lymphocytes. Yeah, you guessed right, as well as memory cells. But yeah, we'll do that in a moment as well. But with proteins, you have certain elements in the molecular structure of a protein, and they include hydrogen and carbon, or hydrocarbon. To be honest, everything in this world is made of carbon. As Walter White said, carbon makes matter matter. Am I muted? Just one second. So you are muted. I don't think I'm muted. I think someone's. Is it back online? Okay, maybe just a mishap. So going back to proteins. Proteins are, yeah, these examples, and they share. uh, there's a couple of elements, sorry, that are found in proteins, and you have carbon, you have hydrogen, you have sulfur, you have nitrogen, and you have oxygen. There was a question on a particular paper, I can't remember what year, and this one, um, you know, a lot of people scored 0 out of 1 because they forgot about sulfur, and that was a bit of a harsh mark scheme point, to be fair. But now you know sulfur is found in proteins. Okay, and yeah, now crucially, proteins they're a chain of amino acids. And amino acids are effectively the monomers, aren't they? Monomers, this is your polymer. They're a chain of amino acids. So if this was amino acid 1, 2, 3, collectively you would make a protein. Now, we understood the elements found in proteins. You also have to work out the tests. So how do you test for proteins? So one question could tie in with kidney function in paper 2, where they mention, the first question could be, for instance, explain why, let's say, why proteins are left in the blood supply. They don't really filter into the BC, the Bowman's capsule. It's because they're too large. And the follow-up could be if the patient enters the clinic and tests for his urine, and it turns out in the urine test there was protein. How would you test for that? So suggest. Well, these are the sort of questions that could come up, and the way you could test for proteins is you could use Biret reagent, okay? And what Biret reagent does is naturally it's blue, but the presence of proteins, like the ones I mentioned here, especially enzymes, is that it goes from blue to purple. So let's go for the purple color here. Don't want to do something in what we call in psychology the Stroop test, where I put the wrong color and the wrong writing. But yeah, so blue to purple. So yeah, and that would indicate there'll be proteins present. Okay, brilliant. So the second one, type of biological molecule I'm going to discuss is, of course, starch. And starch is a type of carbohydrate. And a carbohydrate's in its name. It's got carbon, it's got hydrogen, and 8 in terms of naming chemical compounds. The 8 suffix, the one at the end, refers to anything with oxygen. So like nitrate, iron, phosphate, there's conjugations of oxygen. So yeah, they're the elements. So to test for starch, you would use iodine solution, and just like this font here, so we just like iodine solution. The observation would of course be, um, let's just, it goes from yellow to blue black. So in this case, blue black. We can't really do black. This is just going to camouflage the background, isn't it? Another carbohydrate, a very monomeric, is glucose. Glucose, brilliant. And as you know, glucose is super important for respiration. It's in fact a reactant indeed, and it's a substrate, so on and so forth. Okay. And what would you use to test for glucose? Well, you would use Benedict solution. Okay. I've seen mark schemes where they want you to mention heat, to mention that as well. And of course, the observation observed would be red, blue to red, or brick red. Depending on how much sugar is present, you would have different colors. So I suggest you revise that. Okay, so blue to red, brick red. Okay. The final biological molecule will be on this slide because obviously I've run out. And that ties in, we'll do, actually let me go to the other side. Yeah, number 3 is lipids, fats. And the reagent you would use is two. Well, there are two reagents you'd use. You'd add ethanol and you would dissolve with water. That's crucial as well. Dissolve with H2O, ethanol, this two-carbon figured alcohol. Okay. May as well do a bit of chemistry. So ethanol looks like this. Okay. And those who know, if you know, you know. This is a product of anaerobic respiration, but in plants and yeast. Okay. So here we got it. Here we have it. So this hydroxyl group. And what's the observation, you ask? Well, the observation is the following. The observation is simply um, a milky / cloudy white emulsion. So it's regarded as an emulsion test. This word emulsify, emulsion, we're going to come back to that when we go over the alimentary canal later on in this session. So brilliant. So going once, going twice. So proteins, chain of amino acids, made of the subsequent elements here. Carbohydrates made of the subsequent elements here. Um, Starch and glucose, you have certain reactants you could use, solutions you can use to investigate the presence. Yellow to blue black for starch, or orange-ish to blue black. Glucose, blue to brick red. Lipids, add ethanol, dissolve with water. Okay, observation, emulsion, cloudy white emulsion. Okay, brilliant. Okay, so let's talk about corms. And I have a question in the next slide. Yeah, it ties in with one of the food tests, in fact. The C refers to change. So what would you change in an investigation? Okay, so of course this is the 6 marker you would expect. Sometimes at the end of the paper, sometimes not. And C is in reference to change. What would you change in the investigation? So typically independent variables, what they're looking for. The independent variable is the thing you change. So let's say you're trying to work out the best drug concentration or best drug dosage to eliminate cancer. Okay, so of course, you've got a dosage, different concentrations of dosage. That would be um, your sort of change, the C, independent variable, and the way you could measure the presence, well, the so efficacy, the effectiveness of the concentration of the drug is see how many cancer cells sort of die, get eliminated. So that would be your M. So we skipped a bit. So you would measure. How would you measure your dependent variable? And really make reference to instruments or apparatus. So I would microscopically observe if any cancer cells were destroyed as a result of this particular chemotherapeutic. Okay. If I wanted to work out, let's say on this corner, light, the effect of light on a plant's growth, I would simply measure the height of the plant, the mass of the plant, amongst other things, mass, height, size. But please do not use the word amount. You simply won't amount to anything. I'm reusing and recycling all these bars, aren't I? All these phrases. Okay. CM, yeah, and then the O is something what you keep saying about the organism. So in this case, with cancer, if you are planning to grow your own cells, then make sure they're of the same lineage, if they're the same species of the cells, if that makes sense. If it was about the plant, same species of the plant, same genus / species of the plant, so on and so forth. If it's done on humans, you look at age, you look at lifestyle choices. All of these confounding variables should be kept minimal. Okay, the age should be the same, lifestyle choices, um, diet, amongst other things. Now the R So I need to squeeze an R here. Simply say repeat. Okay? Repeat. Please quantify it. So I say repeat X times. Now, those who know, if you repeat, you would improve reliability. Improve reliability. Okay. So mass, height, size, and then another M for measuring the element of time. So if you want to measure time, and so on and so forth. Okay. And then the S is other sort of things you can keep the same. So really and truly abiotic factors. So going back to this, drug doses in cancer, you could say, of course, the thing you would change of the concentration, you could say the same volume, um, temperature as well in the system, in the actual environment, because that's abiotic, pH, these sort of things. Okay, so yeah, forms. C for change, O for what we keep saying about the organism, R for repeats, mention and quantify how many repeats you do, measure the dependent variable and tell me how you'd measure it, measure the time, and S for same, what would you keep constant, other control variables. And the goal of control variables is to keep a valid, well, to make sure your comparisons are valid. V for valid, okay, validity. So 6 minutes. Some scientists have investigated the effect of vinegar, a weak acid, on the digestion of starch. These design investigations to discover the effect of vinegar on the digestion of starch include experimental details. So really refer to any potential solutions you'd use. Okay? Bit of a tricky question, I must admit, because it ties in with the food tests, especially starch. Okay, and of course it affects enzymes as well. That's it. There's a link. So I'll dedicate 6 minutes on the timer. Good luck, and we'll go through the mark scheme. Okay, guys, another 3 minutes, and then we'll explore the mark scheme. Okay, guys, don't want to waste time. So yeah, so the change in this particular question was that you would use different concentration of vinegar, or you could simply do vinegar or no vinegar. Okay, vinegar, no vinegar. This no vinegar is a control. Okay, so this might be a question saying what is a control in this particular investigation? It's the thing without. If you were doing drug, um, so let's say a drug trial, you would have one group with the drug versus the placebo, meaning no drug, and this whole group is a control. Control, yeah. Thirdly, another example could include if someone had a Alzheimer's brain, for instance, if there's a sketch of an Alzheimer's brain, this could be Alzheimer's, whereas this could be someone healthier. It's got more neuronal sort of integrity to this, whereas this one, atrophy, less neurons. Alzheimer's can be compared to a healthy control, okay, because you're drawing comparisons to it. Yeah, so the bet on controls. So O is for keeping the same mass of the starch, flour, bread, potatoes. So even gone further and mentioned sources of starch. Repeat. Okay. Sometimes in mark schemes they want you to quantify it, so you're lucky if you didn't quantify it in this one. And yes, they wanted you to use iodine to investigate if starch is digested. Now, if starch was digested, iodine would stay orange slash yellow. Okay. If there was no starch, meaning it was digested. Okay. You could even reverse mention Benedict's, because obviously digestion of starch leads to sugars, and sugars lead to the presence of starch. Sorry, the presence of like glucose and other really small sugars. Mention element of time, measure the time it takes for the starch to be digested. So it ties in with rate, and anything with rate is with respect to time. And go further sometimes. Mention the use of stop clock. Okay, we don't know what the mark scheme is going to be like in future papers. Go further, mention the apparatus. Okay. S, what you keep saying, you'd keep the same temperature, use a water bath. So it ties in with the use of abiotic factors, doesn't it? Non-living variables at play here. Use a water bath and so on and so forth. Crucially, same time to react, same volume of amylase. Okay. Um, Same volume of vinegar, same volume of iodine, same volume of Benedict, the ones you used in your apparatus. Okay, that's it. Okay, so now we've gone over. So someone's asked if there was a six marker and I mentioned Benedict's, you would talk about you would mention Benedict's if there was the color change from blue to red or one of those spectrum of colors that would indicate starch has been digested. Okay? If it stayed blue, if it stayed blue, that would mean that starch is still present. Because going back to this slide here, Glucose or other sugars can only be detected based on this color change of red and those spectrums. If it stays blue, no glucose. So in this case, there is starch. I hope that answered it, whoever answered that question. I imagine there's like a 10-second lag. These are streams. Okay, so let's move on. Transport processes. You have three transport processes, and this is between cells to cells. This sort of multiverse of the cell environment. You know, imagine, I don't know if anyone's watched Ant-Man here, but the quantum realm. Imagine that being in the human body from one particular cell to another cell. There's all these processes and proteins and carriers. It is truly fascinating, and one of the processes includes diffusion. Okay, diffusion is passive, meaning it doesn't require energy. Really and truly relies on the inbuilt kinetic energy, but we always regard it as passive. Okay, and this is the movement of substances from a high concentration to a low concentration. Okay, obviously conc is a key for, I meant concentration. I like to abbreviate things, as you know, and shorthand them. Um, so yeah, it's passive process. It's from a high concentration to a low concentration. Okay, examples include oxygen diffusion, where it goes from alveoli to capillaries. Other examples include sodium entering cells and so on and so forth. So that's diffusion sorted. Just like diffusion, this ability of moving from high concentration to low concentration is observed in this process called osmosis. Okay, and but that's only crucial. Well, crucially, it's only tied in with water. It's exclusive to water. And I actually got a osmosis workflow, a way to answer those particularly tricky questions in a couple of slides to go. And with diffusion, there's ways to enhance it, and you can increase surface area. You could reduce the thickness of the wall. You go for thin walls rather than thicker walls, and so on and so forth. Other phrases to include are down a concentration gradient, so on and so forth. Now, osmosis, those who know that osmosis is the process of movement of water, the net movement of water from a high concentration to a low concentration through a partially permeable membrane. Okay. So it'd be an absolute godsend if this was a question on the paper on Tuesday, wouldn't it? Yeah. Okay, and thirdly, the last one includes active transport. Okay, active transport for one uses ATP, energy source from respiration, aerobic respiration, really and truly. Um, does anyone know what is the site of aerobic respiration in the in the cell ultrastructure? Well, yeah, it's mitochondria. Well done, mitochondria. If a cell has a lot of mitochondria, it meets the demand of active transport through providing energy to really drive the movement from a low concentration to a high concentration. Okay, so that's the key difference. Okay, examples of active transport include mineral uptake from the soil for plants. Okay, those who are doing the triple award of the IGCSE course, you know that at the kidneys you have PCT, the proximal convoluted tubule, and all those important substances such as glucose. Well, all of glucose gets selectively reabsorbed through the means of active transport. So those are some examples. Okay. So going once, going twice, diffusion, osmosis, active transport, differences. Well, one difference is diffusion is passive, active transport is active. Active transport uses carrier proteins, that's another one. Okay. High to low, you have the right to know that active transport goes from low to high. And yeah, that's it really. But the osmosis workflow. Now, you're probably wondering what this is. It's not particularly a topic. Workflow means a way to do something in a flow chart. So it's all about the relationship between a solute and water. So if I were to make a graph, okay, and let's say the concentration of water on the x-axis, concentration of the solute, like a salt, for instance. A salt, such as NaCl, sometimes it could be sucrose, a sugar, okay? So if you increase the water concentration, okay, the solute concentration drops, okay? Okay? Now, same case with if I swap the x-axis and the y-axis. If I got the solute concentration and the water. Oh no, I've done this exact same thing. Remove this. If I've got water and solute, if you increase the solute concentration, that goes down. Okay? There's an inverse relationship between these two. But collectively, let's just make it, one graph on its own. So blue for water, red for solute. As you increase the solute, the water is reduced. Potential, if you increase the water, the solute reduces. So they're inversely related. So that's a concept you need to understand. So for instance, if you had concentrated NaCl and you place some red blood cells in there, okay? Naturally, because it's concentrated NaCl, the water concentration goes down. The water concentration goes down. Okay, it drops. Okay? So let's talk about it in this context, okay? What's going to happen to these red blood cells? So number one, in terms of the workflow, you would establish what is higher or lower. So what is higher or lower in water concentration? Now, guys, you can even use the word water potential. So there's a symbol, like the Aquaman sphere, I think it's called. That relates to something called water potential, okay? So you can say water potential as well. It's another phrase. So establish what is higher or lower. And what we're establishing is what's in the cell and what's outside the cell. So in this case, if I draw, let's say cells are here, and in here we have a concentrated NaCl. So let's say, and because of that concentrated NaCl, it has a reduced water concentration outside the cell. And even establish it from outside versus inside, establish that. And secondly, you make a comment on whether water moves in or water moves out. You have to make a decision. Water moves in or moves out. Now in this case, because you have concentrated NaCl outside the cell, that means there's a lower water concentration slash water potential outside the cell, meaning inside the cell, this red blood cell is higher. And please be comparative. So put the ER at the end of those statements, okay? And in this case, water moves out. Number 3 is simply discuss, well, mention this process via osmosis. Just mention that through osmosis, okay? There's a mark waiting for you in the mark scheme, okay? And it's osmosis. So write it down. Number 4 is then discuss the effect of the cell. Now, simply, if the water leaves a cell, it would shrink. Cell will shrink. So in this case, because it would leave the cell because it's concentrated NaCl outside the cell. And in fact, if an influx of water moves in, it would promote lysis, cell lysis. Okay, it would cause it to simply burst. So yeah. So this is an osmosis workflow, and this really can be applied to A level as well. But with A level, you have water potential. Okay, you'd use that phrase. So yeah, that's the osmosis workflow. Now let's reverse it and let's say it's a dilute NaCl. So if I reverse this a bit, invert the arrow going upwards, you have increased water and you have a reduced level of solute, reduced level of NaCl. Then it will be the reverse. Because the red blood cell has a higher water potential slash concentration than the solution, water will leave this other way around. Sorry. If the red blood cell has a lower water potential than outside the cell, water would be drawn in and through osmosis, and then because of water being drawn in, it would promote bursting lysis as a consequence. So that's the workflow. Okay. So yeah, any other questions? Because I've been talking for a while. Yeah, ways to enhance these transport processes quickly. Increase surface area. This would maximize diffusion or active transport, whatever the question asks you. Okay. Crucially, thin walls. And I can tie it in with the alveolus. Have we got a diagram here? Yeah. The alveolar has thin walls. And because of the thin walls, you can have a better oxygen diffusion, can't you? Okay? And they're one cell thick as well. Just like the capillaries are. And as you can see with someone with emphysema, they have a more thicker wall due to the scarring and damaging. Okay. Okay, so thin walls, and yes, the short diffusion pathway can be achieved, or short diffusion distance. Number 3, another way to maximize it is energy, and energy is linked with temperature. Okay, temperature is linked with kinetic energy. So more particles, see more kinetic energy. Of course, for active transport, if you have ATP, as I mentioned, that can promote this process. It can really drive the movement. Okay, make it more active for active transport. Another thing is a concentration gradient. Okay, number 5, concentration gradient. So on and so forth. Okay. Now, we will go through this in the next 5 minutes, but just for 3 minutes because I need a bit of a break, aren't I? Could you fill in this table? Okay. Good luck. 3 minutes, and then we'll go over enzymes. And also, as an extension, can you tell me where the enzyme is produced? Where are these enzymes produced? Okay. And then we'll go over the heart in the next hour, as well as components of blood, as well as the respiratory system, nervous coordination versus hormonal, and conclude with ecology in one slide. Right, good luck. Yeah, even in the next hour, as we're going over the heart, the blood, these are all AQA topics. So if anyone is doing AQA, or if you know any other AQA biologists, please could you share the link, so everyone can benefit from this hour. And also Edexcel, but that'll be more paper too. Edexcel GCSE rather than the IGCSE. Okay, guys. So yeah, not quite 3 minutes, but the right answer for this, something that digests starch, is amylase. Okay, and amylase can be produced by the salivary gland, located near the mouth, as well as small intestine and pancreas. So one of the exocrine juices of the pancreas, the digestive juices, include amylase as well as lipase. And what lipase does, it digests lipids slash fats. And those who answer correctly, it's glycerol and fatty acids. Maltose, it's in its name. Maltase, whatever substrate the enzyme digests, it's in the name. So like protease will be proteins. So that's a mental shortcut. Okay. And if you break down maltose, you'd make glucose as a result. There we have it. And enzymes cause conversion from A to B. At GCSE, you perceive them as being digestive. They break down stuff. Okay. But later on, when you pursue like a biomedical science degree or pursue anything bioclinically or biomedically or biologically related later on after GCSEs, you appreciate they do a bit more. Okay. But at GCSE, you have to appreciate the lock and key method. Now, the lock and key method is well, if I present this is the enzyme, and if I present the lining here, the lining is in reference to something called an active site. Okay. Now the active site is complementary. It's complementary to the substrate. And here we have the substrate. The substrate would then bind to the enzyme active site, and you would form something called an enzyme substrate complex. Okay, and because the active site places, stresses and weakens the bond of the substrate, you therefore make products. So, say little blocks of products here. Okay, and these in turn um, be used for nutrition purposes. Absorbing amino acids, absorbing lipids and glycerol, and all of those important products. Okay, now enzymes link with temperature. Okay, they link with temperature. They link with pH. Okay, so the next, in fact, I'll do it here. So this curve is always important. If they ask you, explain why, explain the results in a particular investigation, let's say yeast, why do you think um, temperature increases from 0 to 37.5, as in why does that increased temperature cause a greater rate? So you would simply say more enzymes receive kinetic energy, and there's more enzyme substrate collisions. Collisions is important, this word, collision theory. It ties in with chemistry, doesn't it? More frequent, successful collisions. Okay, and then past that optimum, this optimum prime, optimal temperature. Anything past that, enzyme simply denatures. Active site changes shape. Denaturation occurs. Okay, same case could be applied to pH, but different enzymes have different optimal pHs. So, for instance, enzyme P for protein, and it is a protein because it's protease. Okay, it optimizes around 1 to 2 pH, or something like lipase. They optimize over like some more neutral slash alkaline conditions. Okay, but crucially, guys, it's anything that's below or exceeds the optimum pH that results into denaturation, causes the enzymes to denature. Okay, that's all it is. Anything below or past the peak, it denatures. Brilliant. So that's enzymes in a nutshell. They're proteins for one, so you use Biot reagent as well, so on and so forth. Okay. So the heart, going through an awful lot, aren't we? So the heart has a lot of chambers. Okay, well, four, sorry. We have the right atria here. Okay, the right ventricle here, the left ventricle here, and in this quadrant you have the left atrium. Okay, now the middle marked X is the septum. Okay, bottom is the apex, and what the heart does, because it has a lot of cardiac tissue, it has muscle to contract, and that contraction, that contractile force, pumps blood to the rest of the body. Okay. Another thing is that left-hand side is more stronger than the right-hand side. It has more thick walls, has more thick muscle. In fact, you can combine them together. Thick muscular walls. This is good because it can generate tremendous pressure. It can generate high pressure, high blood pressure. And that pressure, because one part would be lower, whereas one part would be higher, and that difference, that discrepancy, that gradient causes blood flow. Okay, so you can pump blood. Okay, appreciate as well in blue that the right hand side is deoxygenated, whereas left hand side carries blood composed with more oxygenation with it, in effect. So deoxy blood is more, it's more concentrated with CO2. Oxy blood, the opposite, more oxygen. So you have increased levels of oxygen. Okay. So yeah, bit of the heart. Next, let's talk about blood. Actually, let's think. What should we do? I might skip arteries, capillaries, and veins. Just appreciate the same thing as well. In fact, no, let's go for it. They're blood vessels. Arteries carry blood away from the heart. Capillaries, they're the sort of the interface between arteries and veins and the cells. Okay, just appreciate they're one cell thick as well. Allow for substance to be used and all the waste substances go out the venule end. And veins, appreciate they're thinner. They have thinner walls, carry blood away to the heart, blood to the heart. They have thinner walls, have valves. Just like in the heart, you have the AV valve. Two sets of AV valves here. And you have semilunar as well, which is between ventricle and artery. So you've got semilunar valve. And the whole goal of valves are to prevent backflow of blood. Okay. With arteries, they have a small lumen, smaller smaller diameter in essence. Has thick muscular walls. Significance of that is to withstand high BP, high blood pressure. Also has elastic fibers. Now, can anyone tell me what elastic fibers do? What elastic fibers do is it stretches and recoils. And this allows for the smoothing of pressure surges. So it reduces any pressure surges. Helps maintain the pressure as well. Okay. And it's the opposite with arteries and veins as a comparison. This is with arteries, they have to carry high blood pressures. Veins, they do not as much. So they have thinner walls, less elastic fibers, and valves, okay? Less muscle. So on and so forth. Brilliant. Now, of course, you have these sort of non-communicable diseases. And one example is coronary heart disease, CHD, and examples include atherosclerosis and this, atheroma deposit. In a nutshell, it can also cause heart attack. Okay, and it's the way I want to teach this slide is that you have myocardial infarction. Okay, myo referring to muscle, cardial referring to heart, Infarction referring to blockage. And let's explain this and describe this. So you have your own blood supply for the heart, okay? The coronary artery supplies blood, oxygenated blood, to the heart's muscles, okay, to the heart, to allow it to, cause its contraction, allow muscle contraction. Now, there's a bunch of effects that could occur with these coronary arteries. One is that you can have fatty deposits. With these fatty deposits issued by a very heavy cholesterol diet, it can cause a clot to form, okay? And if a clot forms, that means you're going to restrict blood flow. Restricts blood flow. And if you have a restriction on blood flow, what does that mean? Well, that means there'll be less oxygen, less respiration, less ATP, less energy released. Therefore, the heart cannot respire and the heart cannot contract. Heart cells cannot contract. Okay? And that's it, really. Heart disease. And it's caused by number one risk factors. Risk factors include obesity, high blood pressure. So one of the effects, if there's high blood pressure, is that it can cause a bunch of inflammatory recruits inflammatory cells, and that you have all these white blood cells releasing platelets as well. That's one way of doing it. Also, it can cause damage to the actual integrity of the blood vessel. Of course, the diet, sedentary lifestyle, lack of exercise, all of these things. Number 5 could include obesity, high blood pressure. So, other things like smoking, lifestyle choices. Genetics, it's in the genes as well. So as I draw this double helix, you could have a genetic tendency to develop heart disease. Yeah. So yeah, we'll go over blood in one moment, but could you guys have a go at this question? Or these bunch of questions. So you have, draw Excel diagram to represent the septum. State two differences. The word state, just list. Explain. You need to have a string of answers that link, statements that link to each other. So you have to really and truly tell me the difference and then the significance, the function of that difference. And B talks about factors that could precipitate the following coronary heart disease. Okay, so I will dedicate how many marks for this? 4 plus 3 is 7. 10 marks. So I'm quite short on time. Let's say 8 minutes. And then we'll do the remainder. So blood composition, you want to talk about artery versus the vein, okay? And the differences between the left ventricle wall and the right. Of course, the left-hand side has greater, well, has more muscle and it's more thicker, for one. And then tell me the significance of it as well. And then factors that could increase the likelihood, the risk of developing CHD. Okay, so not quite 8 minutes, but we'll quickly go through the answers. So X is in the middle of this transverse sectional sort of diagram illustration of this heart. Coronary artery contains more oxygen, it's more oxygenated, and has less CO2. And of course, you could do the converse trainers. You could do the vein, okay, vein having less oxygen and more CO2. And like I was mentioning, it's more thicker or thick muscular walls. You can get MP1 and MP2, mark scheme point 1 and 2, if you mention thicker walls. So a bit of a shortcut in that sense. And it can generate more pressure, blood around the body. It's quite similar to a a mock paper question some students have done in this particular stream. Ways to increase the likelihood / incidence of CHD include genetic predisposition, a high blood pressure, a high cholesterol diet / fatty diet, smoking, stress. Stress is implicated with raising blood pressure, um, a lack of exercise, obesity, all of these. Okay, brilliant. Okay, guys, I'm gonna not do digestion because we mentioned the enzymes, but appreciate that you have peristalsis, which are a rhythmic form of contraction that allows a bolus of food, like a ball of food, to enter the stomach. I also appreciate what bile does. Now, bile does the following. Well, the issue is that all the food that comes out of the stomach, the chyme, the juices, all of that acid would render lipase dysfunctional. It would make it denatured. Okay, that's the issue. Because what lipase does, it optimizes at alkaline conditions, around this end. But if there's acid, making the pH much more lower, active sites are going to change. There's not going to be any digestion of fats. Okay. So what comes to the rescue? Bile. Let's know. Let's talk about bile for a bit. Bile is produced by the liver. Produced by the liver, and also bile uh, is stored in the gallbladder. So just behind it. And what its role is the following. It plays a role in emulsification, emulsifies fats. What that means is it increases the surface area of those droplets of fat. And what that means, in a nutshell, is because you have those larger droplets of fat being converted into smaller, is that you increase the surface area, and then that maximizes lipase activity. If you increase the surface area of something, more particles collide, more enzymes and substrate collisions occur, and this is what bile aids with. Okay, and number two is, of course, you need to neutralize stomach acid, and that's what bile does. It neutralizes stomach acid. Yeah, brilliant. So, breathing in, breathing out. Another word for breathing in is inspiration, okay? Or inhalation. You're inhaling air. Breathing out would be expiration, breathing out air. And this is subjected to a couple of muscles. So in the lungs, you have, well, below it you have something called a diaphragm. The diaphragm contracts. Secondly, your thorax volume increases. Diaphragm contracts and flattens. That's just another point to make. It flattens. Thorax volume increases. Thirdly, lung pressure decreases. And that drives the movement. Okay, because go from high to low. And you have accessory muscles and you have intercostal muscles that contract. It's, specifically it's externals, but intercostals are just fine. Intercostal muscles contract. And it would be vice versa. Okay, it's vice versa. You've got diaphragm contracts and flattens, thorax volume increases, lung pressure decreases, so on and so forth. Number one, so relaxes, you just apply vice versa. This. Relaxes. Secondly, it thorax volume decreases, lung pressure increases. And that drives movement outwards. Okay, so, and those relax. The IMCs, intercostal muscles, they relax. Okay, that's done. Now, like I mentioned, whenever they talk about lungs and gaseous exchange, straight away talk about alveoli, okay? And mention these remarks. Increased surface area to maximize diffusion. Thin walls, one cell thick, as I mentioned here. Moist lining. Now, the significance of the moist lining is that gases can dissolve. The dissolved gases. And fourthly, fourth remark would be you have blood supply and you have a good concentration gradient. Okay, so that's gaseous exchange completed. Now, nervous system and ecology on one slide. Okay. We'll conclude this segment with the nervous system and the endocrine system. Okay. Now, nervous system consists of, well, something called a CNS. The CNS stands for the central nervous system. And the central nervous system concerns brain and spinal cord. In a reflex arc, we bypass the brain. This brain is not involved in the reflex arc. So what is involved? A bunch of neurons, a bunch of, well, a couple of receptors, the stimuli, and then the denouement, the conclusion, would be the effector response. So typically, it's the hot object. If you touch a hot object, a sharp object, what would happen? Well, you would withdraw from that, the withdrawal reflex. The first thing that happens is that you have the following. You have a receptor that detects the stimulus. So the stimulus could be a hot object, okay? Temperature. This is the stimuli. This gets detected by receptors. And those receptors establish a impulse in the sensory neuron. And then the sensory neuron then goes to the relay neuron. This intermediate neuron that's part of the CNS, and it synapses with a motor neuron. And after a motor neuron, it then goes to a muscle, This is what we call the effector. And then the muscle would simply contract, move away from the object. Okay. So this would be a really good question if they asked you to describe the sequence of events. You would do the following. But please remember, in chronological order, it goes from S, R, M. I know some of you have got some interesting mnemonics. Just remember, it goes from sensory to relay to motor. So yes, the muscle contracts as a result. Sometimes it could be pain, so refer to pain receptors, and they establish an impulse. Remember, the neurons carry impulses. Okay. Pain, hot, temperature, receptors, sensory neural relay, motor, muscle, and contraction. That moves away from the stimulus. Another thing is, in between neurons, you have synapses. So if this was a neuron, this was another neuron. In fact, if the signal goes this way, and this happened to be the relay neuron, this would be the motor neuron. Okay? And what synapses are is the release of neurotransmitters. Okay, so neurotransmitters, transmission of neurotransmitters, and this is the chemical message really that causes impulses to continue to be established and continue to be firing in a way. So yeah, importance. Well, they're rapid, they protect from harm, so on and so forth. Brilliant. Now, another sort of messaging system is the endocrine system, and this deals with the release of hormones. And hormones are chemical messengers. And what are chemical messengers? They send the message. They're prophets in a way. Send a message from one organ to another to issue a response, to exert an effect. There's loads which we'll go over. Crucially, they're produced by glands. Of course, the master gland is referred to the pituitary gland. Glands, okay, as well as it travels through the blood. Travels through the bloodstream. That's important. And there's a bunch you need to know. You need to know that insulin produced by the pancreas. This reduces blood glucose levels. You need to know about adrenaline, which I made a video about, produced by the adrenal gland. And that increases heart rate, as well as other things. It increases blood pressure, blood flow to the working muscles, causes eye dilation, pupil eye dilation, so light can enter, and you can assess the danger, amongst other things. Number three, another hormone is estrogen. Estrogen and testosterone. Of course, there's links with the menstrual cycle, but just for this sake, we'll just say it's used in female sexual characteristics. Okay. Also, ADH, a bunch of others, but crucially, they send a message, don't they? All these hormones. Drawing comparison between these two systems, yes. So these are a couple of questions that come up as well. Like if you had to describe the difference between hormones and how neurons work, or those systems. One is the vehicle is blood. Neurons are nerve cells and they carry impulses. The nervous system is faster, quicker transmission. However, short-lasting. The effect is short-lasting. Where the hormonal coordination, slower, but long-lasting. Also, not to mention the hormonal system produces, they're produced by glands, the hormones that are. Whereas they're coordinated from neurons, aren't they? The impulses, electrical impulses. The message is electrical, yeah, for one. Produced by glands, long-lasting, slower. They're target specific cells and organs, etc. Brilliant. There we go. So in the next 5 minutes, I'll just get some active recall questions, okay? For those who are tuning in still. I don't know how many people are still watching. But if you could do the following. Discuss the differences between the nervous system and hormonal system, please. Just as a way to memorize, you still retain the information you've learned or revisited in the session. Okay, brilliant. I'll dedicate 5 minutes. Mention examples as well of hormones that you know of. So yeah, 5 minutes. And then we'll conclude with ecology in one slide. And a couple of questions, and then that'll be the end. Okay, so yeah. Examples include obviously the nature of speed, where it's produced, the vehicle, blood versus nerve cells, neurons, electrical impulses versus chemical messengers, long lasting, the effect, all these things should be mentioned and be assessed and assessed. Now, the last one, ecology on one slide, the things I think that will come up on the paper on Tuesday. And we'll start off with abiotic versus biotic. Now, bio refers to life, isn't it? Abiotic are non-living things such as pH, temperature, light intensity. Biotic examples include food, if there's competition amongst species, um, if there's a new pathogen, like the one I mentioned, TMV, tobacco mosaic virus, if there's pests and pesticides, what they can issue. Abiotic versus biotic. And you have trophic levels. The bottom, sorry, is producers, top producers. These are the plants. What eat the producers are the primary consumers. What are on the top are the tertiary. They're regarded as apex predators. So yeah, apex predators. And then, of course, in the middle is secondary consumers. Okay, these are similar to a food chain. However, the food chain looks like this. You could have plant, deer, or fox. Okay, they're shown differently, different representations. And then you got a food web where you have convergence, where a plant might be eaten by different types of herbivores, and different types of herbivores can be eaten by different types of carnivores and omnivores and that sort of thing. Okay, but clearly there is a lot of energy loss, isn't there? The value that's been put across in the biology IGCSE specification is that 10% of energy gets transferred. Okay, transferred from one trophic level to another. So clearly there's a lot of energy loss. So energy loss could be the following, energy loss through active transport. Remember that plants require active transport to have the mineral uptake. Uneaten plants, parts of the plants, or the animal, such as the bone. No one's gonna eat the bones, are they? Because remember, bones have an tremendous amount of energy stored in those, structures. Respiration. Respiration releases energy through movement, through heat. Generation of heat, which produces heat. Remember that respiration produces heat. A lot of energy is lost through feces, so on and so forth. Another thing in ecology is quadrats. Okay, using quadrats, these metal frames. So the first R is you would randomly select these quadrats. Okay, the use of randomization is to avoid bias. Okay, secondly, as you do many repeats, if you have a lot of repeats, you improve reliability, as I mentioned before. Thirdly, count how many species of insects or plants, or depending on the context. Okay, because you have so many repeats, you can calculate an average, a mean, and you can discard anomalies. So that's the goal of a quadrat. A sampling method, a metal frame, and it's a sampling method. Okay, there's a bunch of definitions that I suggest you revise as well. I'm not going to go over it in this slide. I'm just going to go over a few more things like the carbon cycle. Okay, the carbon cycle is anything that releases. We'll do it on that slide in that corner. The carbon cycle deals with anything with carbon and carbon dioxide and glucose. Carbon cycle. 4 processes of interest: respiration, release of CO2, photosynthesis, absorb CO2, number 3 is burning fuels, combustion. Number four includes decomposition, and they release CO2. So on and so forth. Um, yeah, carbon cycle, abiotic versus biotic, these trophic levels. Um, Towards the end, after this question, we'll go over eutrophication, and that will conclude my session. So yeah, and if you can have a go at these questions, 8 marks, 8 minutes. Yeah, explain why only a small proportion of energy contained within one trophic level, obviously that approximately 10%, transferred to the next. Clearly, there's obviously energy losses, and also describe how scientists can compare population size of stargrass in two areas of ecosystem. Obviously, two sets of quadrats, isn't it? Well, two groups with different quadrats in. So yeah. Okay, guys, I'll just do a 3 minute timer. You can just sort of note them down. Okay, um, we'll go through the first question at least. So if you mentioned that not all organisms were consumed, some died, decomposed. Yeah, that's a good point as well. Uneaten, like bones, energy lost through these metabolic events, heat, respiration, muscle contraction. Some things are not digested, which are include feces, excretion, so on and so forth. and then let's give you another 2 minutes to answer the contract question. So yeah, you would use a quadrat, random number generator, count how many numbers of starfish was it? Stargrass, sorry, and there's a kingdoms with it. Ecosystems, count them, repeat, multiply, scale up to calculate the numbers in the area, and that would warrant justify 4 marks. So the last part before the thank you for listening slide, let's talk about eutrophication. Okay. Now, eutrophication is a process by which anything that runs off into the water supply, they include like pesticides and all these chemicals, they can act as pollutants and could encourage the growth of algae. This algae bloom fertilizers can encourage an algae bloom growth. Okay, so this on the top surface of the water. That's an issue because the sun cannot that photon of light cannot reach the underwater plants. Sunlight blocks sunlight, blocks the ability of those photosynthesizing plants to photosynthesize. These plants here cannot photosynthesize. So when they reduce photosynthesis, they reduce oxygen levels. Fish are also affected as well, because if there's less oxygen, these fish here, their population is going to drop as well. So less oxygen for fish, which reduces respiration, which reduces energy, ATP. Okay. Another thing that further depletes oxygen levels are bacteria. The bacteria that decompose, they use oxygen. They're competing, aren't they? There's a competition of oxygen. And we'll do a bit more on this in the paper 2 crash course to come. And I've just got a request now to do genetic diagrams. So in the next slide, we'll do genetic diagrams and I'm going to call it a day. But in the next crash course, we'll definitely talk about fish farming and all of this because they're more triple content. Like for instance, even protein synthesis, all of that's triple and whatnot. So the last one is, let's just make an extra slide, is Punnett squares. Now, really and truly, what is an allele? An allele is a version of a gene. What's a gene, you ask? Well, a gene is a section of DNA that codes for a protein. Okay? Not for a characteristic, for a protein. There are two types of alleles. They are dominant. And a dominant allele is simply an allele, a version of a gene that if that can only express, I mean that always, that's the word you're going for, that always expresses the phenotype if present. So this includes, let's use one example of Huntington's. Now, Huntington's disease is a neurological disease that affects neurons, and it really causes a bunch of problems like paralysis and other sort of presentations. It's caused by a dominant allele, and if you have capital H, which is obviously caused by a dominant allele, it's a key for a dominant allele, then you can either have uppercase H or lowercase h, or double H, double H. Uppercase H and lowercase h, this is what we call as heterozygous. A heterozygous dominant. Now, if the patient has this particular genotype, they have Huntington's disease. And also, if they have homozygous, well, they will have Huntington's disease. Okay? Now, another thing to understand is another type of allele, which is called a recessive allele. A recessive allele can only express the phenotype as homozygous. As homozygous, and the only time it's homozygous, this is like lowercase. So cystic fibrosis. Cystic fibrosis is caused by a recessive allele, the trait's caused by a recessive allele, the phenotype that is, if you exhibit the disease. So the only case you can have the disease is if it's homozygous. Okay, lowercase c. Now, if you have uppercase and lowercase c, this heterozygous, they do not have the disease, do not have cystic fibrosis. But they act as a carrier. They act as a carrier, crucially. Because if mum and dad have heterozygous, and you cross them accordingly, you would have a chance of having two alleles that are the same and recessive, making them prone to cystic fibrosis disease. And let's explore that in this slide here. So Punnett squares, always use Punnett squares. I'm not a fan of genetic diagrams. Just draw this table and represent the gametes. So if mum and dad are this, heterozygous, the gametes will be these. Okay, these are the gametes, and you can represent these gametes in these columns, these rows. So uppercase, lowercase. In fact, I'm gonna make this a bit bigger. Uppercase, lowercase. So uppercase, lowercase. Just make that one super small. Uppercase, lowercase. So they're uppercase together. So this one's not going to have cystic fibrosis. Uppercase, lowercase. They're not going to have cystic fibrosis because there's a dominant allele. Dominant always outweighs recessive. Uppercase, lowercase. So the probability the person has cystic fibrosis is 1 out of 4. Okay. Now, if I see other questions such as if the probability the patient has offspring has cystic fibrosis and is male, so pre the actual mass notation here, set notations, then they're two independent events, aren't they? So you do a quarter times the sex determination probability, which is always a half, as you know, with XY and XX, XY for male and double X for female. Yeah. So, and that of course would be 1/8. So, you got to think of this. The hardest end of questions, they tend to be like the parents always tend to be heterozygous. They always act as carriers, and the disease is caused by recessive. Um, so yeah. Let's see what time it is. 55 past. Yeah, that concludes it. Codominance and all of that stuff come in paper too. Yeah, going one more time. Not for everything, but just like oh, didn't go over blood. We can do blood nuts next time. Okay, this was the sort of program, and we basically covered it. There was blood we didn't. I'll probably express that in a video, all the components in blood and the particular cells. And yeah, there should be enough for your topic 2 revision slash ecology revision. You need to go revisit genetics and use of biological resources. I'll see if I've got any time tomorrow if I'm going to do a crash course again, another hour. But in the meantime, stay focused, keep revising, okay? Be calm, collected, okay? Stay motivated. Do some exercise as well. That's going to promote a healthy brain if you have any time in your schedule, okay? And crucially, just stay positive. You guys can do really well on Tuesday. And yeah, there's still time on Monday, so I'll be at work on Monday if you need any help. So yeah, take care, all the best. Till next time.